Hi, welcome back. Today, we'll be focusing on the topic enzymes. In this topic, you will learn that enzymes are biological catalysts, how to classify enzyme, and characteristics of enzymes. Earlier on, I've mentioned that enzymes are biological catalysts. Before we define the term biological catalyst, let's look at what catalyst means. Catalysts are substances which can speed up the rate of chemical reaction without itself being chemically changed at the end of the reaction. By using a catalyst, you can speed up the rate of chemical reaction, which means that the process of a chemical reaction can be shortened, and at the end of the process, catalyst will remain the same as before, and hence you can actually reuse the catalyst again. An example of catalyst is manganese oxide. Manganese oxide is used to decompose hydrogen peroxide to water and oxygen. Enzymes are biological catalysts. It means that enzymes, they are made of proteins. Enzymes alter the rate of chemical reaction without themselves being chemically changed at the end of the reaction. This means that enzymes will help to speed up the chemical reaction that's taking place inside our body, and at the end of the chemical reaction, they remain the same as before, and hence they can be reused again. Enzyme catalyze digestion process that's taking place inside our digestive system, which will help to break down large molecules into simpler substances which are soluble in water, and hence it can diffuse across the membrane in our small intestine. Enzymes are also used to build up complex substances, for example, process of making proteins by the cells by combining many amino acids together. Enzymes are also used in breaking down complex substances. For example, breaking down harmful substances to less harmful ones, which normally takes place in the liver. In this diagram, it shows that enzymes, for example, sucrase, will digest the substrate, which is sucrose, into smaller pieces, and after the products are released, you will notice that the enzyme active site, which recognized the substrate, remain the same as before, and hence it can be reused again. How are enzymes classified? They are classified according to their chemical reactions that they catalyze. For example, hydrolases, which are commonly used in digestive processes. Carbohydrases is an example of hydrolases which will digest carbohydrates. Now take note, enzymes will always end with the letter ASE. If you see a letter ASE at the end of a word like that, for example hydrolase, you know that this is an enzyme. So a carbohydrate ends with ASE is an enzyme. Example of carbohydrates will be amylase, which will digest starch, and cellulase, which will digest cellulose. Proteases will digest proteins. Example, the pepsin in the stomach helps in digestion of proteins. Lipases will digest fats or lipids. Example, pancreatic lipase, which is found or produced by the pancreas, helps to digest fats or lipids. Enzymes are also classified according to the process they break down. Chemical process to break down the food is by hydrolysis. The word hydrolysis means that this process involves water. The word hydro means water and lysis means breakdown. So in the process of hydrolysis, water is involved in the breaking down of food into simpler substances. So what is enzymes? We know that enzyme is a biological catalyst, means that enzymes, they are made of proteins and they help to speed up a chemical reaction that takes place in our body. Okay, proteins in nature provides the reactants with more energy than the activation energy. Some chemical reaction that takes place involves a lot of activation energy. Activation energy is referring to the energy that is needed to drive a chemical reaction. With the presence of enzymes, it helps to lower down the activation energy. Hence, reaction will occur at a faster rate. Enzymes are highly specific, and at the end of reaction, enzymes are not altered or destroyed. To summarize what we have learned so far, enzymes speeds up chemical reaction. Example, enzyme speeds up digestion process that's taking place in the digestive system. An enzyme is highly specific in their reaction. For example, a carbohydrates will only digest carbohydrates. Proteases will only digest proteins, and lipases will only digest lipids or fats.